Okay, so I just want to wrap up a little bit some of the things that I discussed with you today. A couple of slides, so that will be the final part of my formal presentation today. And then we'll run into two things. One of them will be to have our colleagues to tell us what you have found in these two papers that we gave you yesterday. And then number two, and this is very important, I would like to organize with you a bit about the assignments of different papers. Well, I know that there are, I counted in the list of participants that I could not have until today. I counted 37 participants. Obviously, we don't have 37 people here. And I don't know, we have to discuss how we assign different homeworks to different participants and so on. So I prepare some papers that I will assign for you. Asmita? Yeah. Do we have tomorrow morning?
If you want, we can do tomorrow morning.
Well, you know, what I was planning was to leave some homework for them to do, but I don't think that we can do much tomorrow because they have to go through the papers and everything. Okay. Because the idea would be on Saturday morning to have these kind of seminars and so on, so, but they have not read the papers yet, so I don't think they will have time yet.
Okay, so we do afternoon like today, tomorrow also?
Well, tomorrow is Saturday, right?
Yes.
So, but you know, you have to explain me. So Saturday is a working day here?
It's flexible, if you want.
Well, I don't know. I have to ask you as well. So I don't know what is the standard. I mean, do you guys work in Saturday morning? You do or you don't? Yeah, you know, I don't know. Will you show up? I mean, do people show up in Saturday morning?
If needed, yes.
I mean, you put a gun in the chest, obviously they will come. But
so it depends on your.
Okay, so I think that for tomorrow it will not be necessary. Okay, so I think that maybe next week we'll have a more intensive, and mostly the last weekend, that is the one that everything will be concentrated. But I think that for tomorrow is too early to have a Saturday session.
Tomorrow is full day homework.
Yes. Okay?
Sure, fine.
Very good.
Online people should also know that.
Yeah. Okay. Okay, great. So then as I told you, I want to organize these assignments and some homework. And I don't know, because we have nominally 37 people, and well, let's discuss that a bit later, okay? Now let's get done with this part. Okay, so in summary, the whole story today was about this gene expression flow, transcription, translation, all the details and all that, and how also some aspects of transcription and translation can be leveraged to do something different from the perspective of synthetic biology, in particular these logic gates that I told you about, and also this idea of using alternative genetic codes and alternative information carrying molecules and all the rest of it. We have discussed about devices, about operons, so I think that we have covered a big background to really move on into the subsequent steps of the program. Okay, so I just want to make a little remark about the ribosomes. So the ribosomes are the most important machine that a living system has, because it's the one machine that decodes the information. That's is a amazing molecule. So as you know, it's composed of the subunits and many different aspects and so. And but to me, you know, every time I look at the ribosomes, I really get amazed of the ability that the ribosomes have to do what they do. So it's just completely amazing. Okay, so the ribosomes have received recently a lot of attention. Now every detail is known, and there are many, say, features that are being discovered in the last few years, and it's still a molecule that is very, very surprising. So you know that the more we know about biological systems, the more complexity we find. For instance, there was this idea that you know, all ribosomes are the same and they all transcribe RNA regardless of the sequence and so. And now there's this growing evidence that specific ribosomes will be interested in some mRNAs as compared to others. So there's that some kind of speciality in terms of the ribosomes, and also some antibiotics that inhibit ribosome action will inhibit only the action of the ribosome in specific genes, but not in every gene and so. So I think that there's still quite amount of knowledge to know about, to understand about the ribosomes, to really be able to use them properly in synthetic biology. Okay, so I just want to share with you this movie that I find quite fascinating. It's a simulation of how a ribosome works. Maybe the definition is not fantastic, but okay. So these are the loaded tRNAs are here. Okay, this is the RNA that goes to the ribosome, and then to get in a row here, and then there's this factor that kicks in, and then the upper part of the ribosomes of the tRNAs goes inside to get the reaction for production of amino acid of the protein running. Okay, you can go faster. And then, you know, in the other side of the ribosome, you have this kind of production of the protein sequence. You know? Well, this is a simulation, of course, but chances are that this is what really happens. You know? And to me, this is very, I mean, amazing. You know, in the sense that you have this chemical machine that is able to do all these things at the same time. It's really fascinating. Okay, so I just want to finish this with this slide that is connected also to the origins of the ribosome. And you may say, well, how come that this super complex machine came to existence? You know, how was evolved? I mean, what were the precursors? So the origins of the ribosome has remained a mystery to this day, you know, because it's not that you have something that is simple and then it gets more complex and more complex and more complex, and you can identify all the precedent steps. In this case, you have something that is already very, very complex. So in the last years, in the last couple of years, there have been a couple of papers or some papers indicating that the core activity of the ribosome is this motif in the whole structure here, that is the so-called the peptide transferase center. Okay, there is a pocket of RNA in which amino acids come together to form complex proteins. Okay, so this is, you know, a ribosome is a complex between small RNAs and some so-called riboproteins, and well, they form these structures and so on. But then the core, the absolute core of the whole thing, is this structure here, and this structure is kept in all life forms. Okay, so if you want to go for a absolutely conserved sequence in every biological system, this is where you have to look at. Okay, in this particular point here. Okay, so this is something quite remarkable. Okay, so just leave you with that idea that ribosomes are very complex, and even the best engineer to this day would be really difficult for him or for her to figure out how a ribosome works on that. Okay, so now let's go to this paper assignment discussion. So I got from Ashmita a list of participants. I don't know how many are online, probably none, I don't know, or maybe some of you, I don't know. And my idea was to every day, because at the end I want every one of you to have the chance to you know, come here and to give a presentation about one paper. And but the fact that the ones that come here face to face are way less than the ones that are registered makes me think that I'm not sure how to organize it. So maybe we should organize the assignments today on the basis of those of you who have come, and maybe subsequent days we can do it differently, right? But you know, I would like to spend every day at least 1 hour of the whole time having presentations of you in of the different assignments, and also discussion. Okay, so far we had very little discussion, and I don't like that, you know. So I think that you should overcome this imposter syndrome that I told you the other day, because when I talk to you, you know, you guys talk very well, and you tell me about your projects and your ideas and everything. But then if I ask you, let's have a general discussion, then you get very shy and you don't say anything, right? So I think that we have to overcome that and engage into interesting discussions. And in fact, today I will raise some issues that perhaps I would like you to have your feedback. You know? But in any case, so my idea was to have a paper. I mean, to have a team of 4 people, and then to give a paper to each of these teams, and then next day, I mean, to have as a homework. You know, all of you read all papers, but the team that is assigned one specific paper is the one that has to make a presentation about that particular paper, going into the reference, the paper, the literature, making some slides and so on. So at the end, you have a kind of formal presentation on a kind of journal club on what the paper is about, what are the plus and minuses, and you have like the better and the worse, right? Okay, so I'm sorry that I don't remember, I don't know your names yet, but maybe this is the time that you tell me your names, and perhaps I can tick here in my list who you are and so on. But I mean, how do you like the idea that I tell you some of the papers that I thought that would be interesting to discuss, and then well, I have more papers than teams that we can make now, but you can make your pick and perhaps to distribute the different assignments to different teams. I don't know. What do you think? So how many are we now are today? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. We are 16. 16 would make 16, like four teams. 4 teams of 4 people. Okay. Let's do that. Okay. I had prepared 6 papers, so I think that we will cover all the papers, and we keep the other 2 for future. So my idea would be to propose 4 papers every day. Okay. So on Monday you have 4 more papers and so on. But just the idea, as I said, is that you all please take a look to the paper. You don't have to read, all of you, every paper in detail. The one that you have to read in detail is the one that is assigned to your group, and then you decide who in the group is going to make the presentation. And as we will be together for some time, you know, every time, every kind of round of presentations will be presented by a different person, okay? So sooner or later, all of you will have the chance to come here and make your real show, okay? Okay, so I'm going to tell you the papers that I selected for this particular case and why I consider that they could be interesting to really go through in detail. So the first paper is this one by Chris Voigt and Adam Arkin on integration by a modular AND gate. This is the full explanation of the part of my talk where I explain this idea of making an AND gate of the basis of combining the T7 polymerase with a suppressor of stop codons, and so on and so on. So that's something that I think is worth to really go through in detail, so you kind of understand the logic and the intention of the authors to do that. Then there's this other paper. I mean, believe it or not, this paper is a paper in Nature, so it's not a cartoon, but it was accepted as it was a kind of iconic publication because it was written by someone called Drew Endy, and it's an explanation of how the biological gene expression flow can be domesticated by engineers to produce circuits and to produce something that resembles the activities that engineers normally do. So, I mean, obviously, from the point of view of reading it is much easier or more fun, but the contents are quite heavy, okay? So don't think that because it's produced in a kind of cartoon fashion, then things are easier. One detail that I want to mention at this point in connection to this paper is this idea that is becoming very popular in the synthetic biology community of, I don't know if you have this discussion here in IIT, but I don't know if you ever heard of the, please tell us what it is.
So it is something like something in the form of a game that when you do some stuff, you get some rewards in return. Then you virtual rewards, but you get some rewards in return. So in many cases, this is generally used to motivate people to do something or nudge them towards doing something. I know this more in terms of marketing stuff. I don't know how this is used in biology.
Yeah, okay. Okay, thank you very much. This is exactly. So the idea, as your colleague said, is that sometimes there are contents that are really, really heavy. If you go to the first part of my talks today, transcription, open complex, that's a pain in the neck, let's face it. You have to study, you have to remember, and it's not nice, it's not really nice, right? So if you could learn that through a method that involves some kind of fun, then the experts in education say that you will never forget things. So if you learn something through studying, I'm sure that in 6 months you have, 6 months, maybe in 2 weeks you have completely forgotten everything I told you in this course, right? But if you learn things through gamification, then you will remember that for the rest of your life, right? So there is this kind of idea in synthetic biology to bring what your colleague said about converting or translating the narrative of deep scientific questions that are normally described in a very rigorous and very serious language into something that is fun. And by making it fun, then chances are that you will remember it much better. So if I give you a lecture formally, what is POPs on what is translation, and with this, as I said today, chances are that I will forget very quickly. If you go through this thing, it's not a game because I'm not making a game among us, but the kind of style of telling the story in a funny fashion with the narrative of a comic, then probably will make you remember some of the concepts that I have discussed today much better. Okay, so this is something that you will be fun. You may have fun reading if you have the chance to do it. Okay, then there are 2 other ones that I suggest you to take a look to. So one is a paper that I mentioned on passing yesterday, but I noticed that all of a sudden you kind of open your eyes like something interesting. And it's this idea of using of programming bacteria to deliver and to produce at the end self-dyeing textiles. And this, remember, I told you yesterday about having these biofilms that can be converted into leather, and then given that the biofilms are genetically programmable, then you can introduce genes for production of dyes or different colors and so on. So at the end, the leather and the textile that you produce happen to be color as well. So that's something that is something. Okay, one thing I have to say about this paper is the following. This is not a real paper. This is a preprint. Did you ever heard about this preprint story? Yeah? Please tell us what is a preprint.
Before going for a publication, they used to publish.
Don't tell me, tell your colleagues. I know what is a preprint. Tell your colleagues.
Before publishing some scientific findings, they just go for preprint, like before having some preliminary results, so that someone might in the similar fields can work and publish that. Am I right?
Well, you are right, but I think it's a bit more than that. So a preprint is, as your colleague said, is a new way of publishing scientific results before they get accepted and published in scientific papers, okay, in scientific journals. So there are various aspects to the things, and the difference between a preprint and a scientific paper is that scientific papers, once they are published, they have been refereed, and I don't know how many of you, maybe some of you have been through the pain of having to publish a paper, but typically you make some work, you may have good experiments or good results and whatever, and you write a paper, and then you send it for publication. And then it goes to reviewers, and then the reviewers in general will kill you. Will say, well, this experiment is wrong, this experiment needs this control, this experiment that did it. And then the editor can say, well, the results are all right. If they are horrible, the paper is rejected, but in many cases say, well, the results are okay, but you need to do this and do that, just follow the instruction of the reviewers, and you have to make a revision, and then you have to make another revision, and this and that. And then during that period of time, maybe some of your competitors would say, aha, this is interesting, we have to get there first. And there are ways in which you can accelerate the publication of something that you are doing. So one of the benefits of the preprint is that even before the paper is formally accepted, there's an evidence that you did that before. And this is a way of what is called scooping protection. So scooping, you know scooping, right? That someone, you are running and then someone goes and go faster than you. So this is one of the most horrible nightmares in research, that you are working very hard in your project and all of a sudden you see a paper of someone, I don't know, in South Korea that has made exactly the same thing that you have done, much better and much faster. And then you are about to commit suicide if you say that, right? Okay, well, in any case, so by publishing a preprint simultaneously to submitting a paper, then you know, you make sure that you establish that you have been doing that work before the other. So that's something, you have priority, it's called priority. On the other hand, you know, you expose your work a bit ahead of time, so sometimes it's not a good idea and so on. And then in the publication business, maybe, I don't know, did any of you go to this meeting that took place a couple of days ago, this EMBO visit? Okay, so I don't know if you remember the guy working in EMBO reports or EMBO journal at the end, saying that maybe in the future, the preprint will be the standard way of publishing things. So instead of sending a paper to EMBO journal or to Journal of Molecular Biology or something, you put your work in a preprint repository, and then it will be reviewed as a preprint, and then different journals will look at it and will decide whether they're interested in your paper or not. Whether or not that will work, I don't know. Well, in any case, so what I wanted to tell you is that this gentleman here, Tom Ellis, has put this as a preprint. So if you make the choice of focusing in this paper, just keep in mind that this is not a fully refereed paper, it's just a preprint, okay? But still, I think it's very good, and Tom Ellis is a good name in the field of synthetic biology and a bit of a competitor of us in this field of leather produced by bacteria. But anyway, it's very nice. Okay, so there's this review that is made by Rafael Silva and myself time ago, and where we review all the, or many of the things that I have been discussing today about logic gates in prokaryotic transcriptional regulatory networks. In this case, it's not so much about engineering new regulatory tricks or logic gates, but about looking at existing regulatory networks and then deciding whether one specific regulatory configuration responds to a logic gate of a specific type, okay? So I think it's a good reading. It's not a paper, it's a review, but I think that we cover many of the issues we have discussed today, plus many others, and give specific examples of different scenarios where logic gates or whatever appear in natural regulatory networks. So that's someone that is quite good. Okay, then there are two papers, both of them from Jason Chin's laboratory. One of them is about the synthesis of an E. coli with an entirely recoded genome. I think that this paper, from a technical point of view, is super interesting, because to assemble a big chromosome, I mean, you cannot synthesize the whole chromosome. You have to go pieces by pieces, and then to create a scaffold where you can put the pieces in order and so on. So technically, it's super interesting. And even though I will discuss in some detail in one of the subsequent classes the organization and the different methods for genome assembly and genome engineering, I think that this is a good aperitif of what will come later in the course. And this is a more recent paper by the same laboratory explaining how codon reassignment enables resistance to phages. You know, well, this other thing, polymer synthesis, you can ignore that. This is the title of the paper. But the idea in this case is to show that effectively the E. coli cells were a degree of genome reduction and genome recoding has been executed, now become very resistant to viruses. Okay, so and I think that the two papers are very nice. Okay, so that's one thing. The other thing is that I send you an email to all of you. I hope you receive it with access to a Dropbox file that is in my computer, where you can download all documents connected to this course. Okay, so I have uploaded there the two presentations of yesterday and today, and also there's a special say file with all these papers that I have listed there. Okay. So I listed 6 papers, and we just need 4 papers. So which are the 2 that you may not be as interested? Okay. I think that all of them are quite cool. So, but okay. So maybe I should skip my paper. So that's because it's a review, it's not a real paper. So and this is like 2 features of the same phenomenon or the same work. So I think that we can group up. So I would propose that we take 1 paper, 2 papers, 3 papers, and 4 papers. Okay, even though this would be 2 papers instead of 1. Okay, is that fair enough? I don't see yeses or noses or noes. Okay, good. Okay, now, how do we organize the groups? Do we organize them randomly, or do you prefer to have a specific, say, convergences with some of your colleagues there? How do you prefer? We can do it randomly, maybe it's better, I don't know. So we don't have any bias, or you prefer to work together with colleagues that you know more? I don't know. That's what do you prefer? Okay, please, let's make like a survey. We do it randomly. Raise your hand. Okay, okay. Most of you. Okay, perfect. Okay, so then let's suppose that you are sitting now randomly. So I can go 1, 1, 3, 4, or okay, let's do that. Okay. Or
I guess you have 14 people.
the what?
You have 14 people, not 16.
16. How many?
14, because I don't think the streaming people will be interested in doing a science.
Yeah. Well, I'm not counting the streaming people. If they don't show up here physically, it will be very difficult to. But I counted.
I think there are 14 people.
But we counted 16 here in this room, right?
Right now, okay, 2 wait.
Or 2 left.
So there were 16 in the morning. I think right now there's 14.
We are 14 now.
Yeah. Sorry, the first session.
Okay. So what do we do? So okay, we can have less papers, or we can have a group of 3, because it will be 3. No, it will be. Yeah. Okay. So we can have 4. We can have 3 groups of 4, and the other with 2, or 2 groups of 4 and 2 others with 3. That would be the bottom line. Okay. Very good. Okay. So let's start like that. So 1, 2, 3, 4. Group number 1. What paper you want to take? Okay, let's do it differently. Okay, I'll give you a few minutes to think about it. But well, you didn't bring your computer, so you cannot really have access to this thing. Let me see how can we do it. So, but just remember, you can make a note that is recoded genome, self-dying textiles, environmental cyanide integration and gate, and adventures in synthetic biology. Okay. So all these papers are in the Dropbox document, in the Dropbox file that you can access through the link that I gave you before. Okay. Okay, I'll do it. Okay. No. Look, let's do it differently. So this is a bit complicated. So I'm going to tell you what paper you will have, okay, period. And then some other day we do it differently, because this will be a bit complicated. Okay, so 1, 2, 3, 4, you go for this paper, number 1, okay? 1, 2, 3, 4, you go for this paper. Okay, 1, 2, 3, then you go for this paper, for the textile paper. And 1, 2, 3, okay? You go for these 2 papers. But these 2 papers are basically the same story, okay? So it's not that I'm overloading you with work, okay? Is that fair enough? Okay. So your work or your assignment would be to, I insist that please all of you read all papers, read them. But the team that we have specified are the ones that have to read that particular paper in more detail, and then to make a little presentation with slides and everything on just to describe the contents of the paper and to have some little conversation and some discussion on whether you consider that paper to be good or to be bad, and what are the holes, what are the benefits, and so on and so on, okay? So let's do that. And if you have difficulties to find these papers in the Dropbox, please let me know, and I will, well, I think that you have all received my email, and you can contact me with no problem, okay? Okay, is it clear what you have to do? Is it clear?
Is it like one of us from 4 has to present? Or
Yes, you decide, you make a little conversation, and then you decide which of you is going to present that paper. But keep in mind that sooner or later, all of you will talk, okay? So it's not that you have to do. It would be nice if the one that is going to make the presentation sends me an email telling me the name of the person who's going to make the presentation and the names of the others, you know, because I think that it would be ridiculous that now I go one by one. Sai Saranga Das, are you here? So it makes no sense. So it's better that you send me an email. You have all my email with the names of the different teams and the name of the one that is going to make the presentation on Monday. Okay? Is it clear what you have to do? Clear? Okay, very good. Excellent. All right, so we come now to the point of having this little presentation by your colleagues. So I don't know which of you want to share your say investigation about the 2 papers that we discussed yesterday. Okay. So you want to you have something to upload in the computer, or you just what? Okay, very good. So, yeah, you can use your computer or you can upload them in mine, whatever is more convenient. Okay.